If my dog barks again, I apologize in advance. I usually have this room closed off, so every time I open it back up, she gets excited for a while that she gets to see them. So, um, hopefully she's done. All right. So you made it through module one. Um, and as I told my classes, module two is honestly the one that scores the highest every year, and where students get I'd say like three-fourths of their points, or at least a good chunk of their points from, just because the questions seem to become a lot more straightforward. There's a lot less they can ask that's really off the curriculum. For the module one part, they often throw in more examples and real life scenarios, as I was told. Um, because of the content, it's easier to do that. Whereas with this section, it's not as common that they throw in all of those examples that may or may not have been talked about in classes. So this one should be more straightforward. I'm going to do my best to get through, especially some of the harder units, but without, again, taking a million years to do so. Um, so let's get started. All right. One of the bigger units, I'd say, that's on the Keystone tomorrow is your cell growth and reproduction unit, which is mitosis, meiosis, and the often forgotten about binary fission. So I'm actually going to start down here with binary fission. Ugh, maybe I just won't zoom in. Okay, so down here in this box at the bottom, we have a image to the right of the box that shows a bacterium splitting. Bacterial division is much simpler than a eukaryotic animal or plant cell division because they don't have as many organelles. They really just have ribosomes and the DNA. So obviously their DNA still gets copied, but there's no nucleus around it. So once it's copied, it kind of can just separate. The cell elongates and it ends up splitting off. This again is called binary fission. I always say binary starts with a B, bacteria starts with a B, so that's who does binary fission. This is also just another word for the type of asexual reproduction that they are doing, where again, they just clone themselves practically. So, asexual reproduction for prokaryotes. Now, for eukaryotes, they split for multiple reasons. That could be to just create more cells, grow and develop, which is what we're going to talk about with mitosis. Whereas then for the reproduction side of things, that's when we're going to encounter meiosis. So before I get into that, we do need to go over the cell cycle first, which primarily focuses here on interphase. So interphase, remember, is about 90% of the entire process. It is the time where the cell is preparing for that actual division. So interphase is split into G1, S, and G2. G1 and G2 are pretty similar in what's happening there. So that's why both of those are colored blue. The S phase or synthesis phase is probably the most important phase because this is where the DNA is replicated. So this is where we're going to take our DNA and completely copy it so that we have two copies. In G1, the cell is mainly just growing, and the cell is still functioning during this time. It's not like the cell is inusable or it's not doing its normal job. It's still able to do what it's supposed to. By the time then the DNA copies and it gets to G2, if the cell needs to grow a little bit more, it will. Not listed here, but often when it's mostly happening is the um, organelles are copied during this phase as well. All right. That then would lead you into one of your two phases. That would be mitosis or meiosis. And for mitosis, let's see if I can zoom in without this fully going away. There we go. Okay. For mitosis, you have your four stages. You have PMAT, or prophase, metaphase, anaphase, and telophase. Lots of songs that are on YouTube for this. Look them up if you want to watch them again. But I'm not going to spend the time right now to do so. Um, for prophase, this is where the nucleus itself is going to break down. So that purple part is your nucleus. And then your DNA often looks a lot like it does in the bacteria down here before prophase. Is in prophase that it's going to spiral up or wind up to create the chromosomes. So the chromosomes here are separated by copies. So one half of each X is one copy, the other half is the other copy. So that is when we get the term sister chromatids. It should be twin sister chromatids because at this point they are identical to each other. Each half is. Spindle is also going to form. That is just the fiber that's going to eventually help separate those chromosomes. The two easier ones, I think, come next. So first we have metaphase. So metaphase, M for middle. They are lining up in the middle of the cell. Keep in mind, again, it's just by sister chromatids. So you have a single X in each location going down the cell. When they line up, the spindle fibers are going to start attaching to the centromere in the center here. So that for the next phase, anaphase, A for away or A for apart, the sister chromatid pairs are going to be pulled apart towards the opposite ends of the cell. This again is ensuring that there is one copy of each chromosome on each side. That way, in the end, they're going to be identical to each other. Finally, in telophase, the chromosomes are going to start to unwind back into the spaghetti-looking shapes that, oops, you saw in the bacteria. I'm just not going to get to it. The nucleus reforms, and slowly the cell is going to start to split. But it does not split entirely during telophase. So one common misconception is that mitosis is the division of the cell. The mitosis phase is not where the cell actually divides, it is where the nucleus divides. Where the cell divides is during the last very quick, very small step, which is cytokinesis, which literally means cell splitting. So this is where it's actually going to pinch off and the process is complete. For mitosis, your cells start diploid, which means they have two sets of chromosomes, one set that came from the mom, one set that came from the dad, and they're going to end diploid. We are trying to create cells that are identical to the original cell before it split. So diploid before, diploid after. And again, this is for growth and repair of damage for organisms. All right, the more complex one. This is going to be difficult to pull up. It's just going all the slides. Okay. So meiosis is, again, to create gametes. So gametes are your sex cells. Those are your sperm and egg. And remember, meiosis is going to go through PMAT twice. So when you are identifying a phase in meiosis, you need to put a 1 or a 2 identifying which step of meiosis you're in. So our cells in meiosis start diploid again, meaning they again have all of the sets of chromosomes, one from mom, one from dad. When they line up this time in prophase, where they start to pair up, that is going to now be in groups of 2 X's, okay? So before, remember, we just had them as singles. They were all separate from each other. But now our homologous chromosomes are going to pair up. Homologous means that they are the same chromosome, okay? They have all the same genes. However, the one's from mom and one's from dad. So the genes might have different versions on them. So here you can see, let's say the darker ones are mom, the lighter ones are dad. Okay, so you get one of each copy. Now, the reason there are four sticks instead of just two is that we still had to copy our DNA. So you're still getting a copy of each homologue is called before they pair up. The most important thing that happens in meiosis, I'd say, is in prophase one, which is the crossing over. So you can see here that they're kind of like crossed over each other and they are trading that section of DNA. This is a random process, but the DNA that is traded will be the same section of DNA on both chromosomes. So if it's trading for the gene eye color, it's also getting the gene eye color back, just the different version of that trait. Again, everything else that happens in prophase also happens here. So the nucleus is going away and spindle is forming. In metaphase one, the homologous pairs line up. So again, we are not in a single file line where each one lines up in the middle. We're kind of getting the pairs of two to line up in the middle. Anaphase one, we're splitting an entire X instead of just the single side. Okay, so we're not splitting up sister chromatids yet. We're just splitting up the homologous pairs. And then in telophase one and cytokinesis, same thing. Now we have one set of the chromosomes in each cell. So this is when the cell actually becomes haploid because we now only have half the number of original sets of chromosomes. So in human cells, we start with 46, and at this point is when you actually have 23 chromosomes left. The problem is we might have 23 in each cell, which is great. However, there are still copies, okay, of chromosomes in each of those cells. So we now need to split up the copies, which is why we do prophase 2. Prophase 2 looks almost identical to mitosis, except it's happening in two cells at once, okay? And instead of the cells being identical at the end, they are all going to be genetically different because of the crossing over that occurred. Okay, so prophase 2, metaphase 2, anaphase 2, and then they split, and you get four genetically different haploid cells. It's a mouthful. Okay, so that's meiosis. Main thing to keep in mind is the homologous pairing and the crossing over in that first meiotic division. One more thing with mitosis and meiosis is the non-disjunction. This is the process that happens when chromosomes do not separate correctly during anaphase, at any stage of anaphase. So if they did not separate, okay, you're going to end up with the wrong number of chromosomes in both cells that are formed. One of those cells is going to have too little chromosomes, and one of those cells is going to have too many chromosomes. Okay, so every time this happens, two cells are affected, maybe more. So this is when things like Down syndrome or occur because that is an extra chromosome 21. So an extra 21 was pulled over during anaphase, and therefore it was passed on that way. Okay, so another big thing I would know for mitosis and meiosis is the differences that I was trying to hint at as we went through. So again, meiosis goes through I don't know what I was saying there, two nuclear divisions. So PMAT twice, whereas mitosis is once. Meiosis is creating gametes, so your sex cells, whereas mitosis is your somatic cells or body cells. Think my toe is a part of your body. The toe is a part of your body, at least. Okay. And therefore, it is producing those body cells. Meiosis produces four haploid daughter cells, so only one set of chromosomes, whereas mitosis is producing two diploid daughter cells, so identical cells that have two sets of chromosomes in them. So the total number for humans is 46. And then meiosis, genetically different, mitosis, identical. Alrighty. I think that was probably the most complex one for us to go over. So now we're going to get into the molecule that's so important to split during mitosis and meiosis, so DNA. Um, so DNA, remember, is a double helix, okay? And that double helix is bound together in the center with hydrogen bonds between what are called our bases or nitrogen bases. So we have our adenine, thymine, cytosine, guanine, and then when we get to RNA, uracil. So when DNA replicates, the first thing I want to talk about is that it is considered semi-conservative. So we have our two strands here, and you can see that they are separating apart from each other. Eventually, they're going to fully separate so that the two red strands are no longer connected. One of strand each, as you can see in the diagram to the right of the original model, is going to receive a red strand, and then it's going to receive an entirely new strand. So it's going to receive the blue as it's showing. Okay, so because of this, DNA replication is said to be semi-conservative because you are saving semi, half, and conserving means saving. Okay, so you're saving half of the original model or molecule, and you are getting a new half. So half old, half new. Remember with the bases then, it is important which base goes where because A and T can only bond together at all times, and C and G can only bond together at all times. So we're going to get into genetics, and then we'll eventually get into the protein synthesis. So the image here, remember DNA makes up chromosomes. Okay, so chromosomes are just wound up, organized DNA. And that's because we have a lot of DNA. There is a lot of complexity. There she goes. Pause for 2 seconds. But I'm going to mute myself to tell. Okay, I think I stopped her. So what was I saying? We have a lot of DNA. Okay, so we need to keep it organized in the best way possible. So when we have our DNA, again, you are going to have DNA that's from both parents. You inherit both. And that's when on our chromosomes you get what are called alleles. So these are versions of a trait. They could be the same, okay, if your parents have the same trait, or they could be in most, like they are in most cases, different, where you're going to get a different version of the trait from each parent. So again, these are called alleles, and our alleles are often classified as dominant or recessive. Okay, so a dominant allele is represented with capital letters, whereas the recessive allele is represented with lowercase letters. So when we start looking at genetics, there's two main vocab words here. We have genotype and phenotype. A genotype is explaining the combination of alleles using the capital and lowercase letters, while a phenotype is the actual physical characteristic that's shown. So that could be color, like it's showing down here. It could be height, it could be wrinkled versus round, anything like that, that is the actual physical characteristic. So when we're looking at genotypes, the three possible options are homozygous dominant, homozygous recessive, and heterozygous. So remember, homo means same. So homozygous, same, and then dominant, I already told you, is capital. So that gives you two capital letters. Homozygous recessive is, again, homo, same, recessive, lowercase, so you get two lowercase letters. Heterozygous, hetero means different. So with hetero, you are getting one uppercase, one lowercase. With normal genetics, you are going to have what's called the law of dominance, where the dominant trait is going to overpower or hide a recessive trait. So if you have two dominants, obviously you're going to get the dominant trait because that's all you inherited. But if you have a dominant and a recessive, you are going to show the dominant color, which in this example is a purple flower. So even though that flower inherited the white trait, it is not going to show it. Whereas obviously then, if you get two recessives, you're going to get that white color. Next page. Okay, one way you can predict the offspring of a cross between two parents is through a Punnett square, as I'm sure all of you probably remember doing. If you don't, you probably weren't here. So Punnett square, remember you just put one parent on the top square, one parent on the side. Okay, so it's not like here, or I saw students trying to put like capital lowercase on just this first box. Okay, so one letter for each box. Okay, and then you just kind of meet in the middle. So capital A is over this box. This capital A goes here, lowercase drops down. Capital drops down, lowercase gets pulled over, and then lowercase in that one. Okay, and then that can predict that the example they have here, you have 25% chance of homozygous dominant because there's only one with two capital letters. You get a 50% chance of heterozygous since there are two boxes, and you get a 25% chance of homozygous recessive. So remember, each box is worth 25%. So if there was none that were lowercase, that would be 0%. If there were three, if it was just asking which ones would show the dominant trait, it would be 75%, and so on. Okay, so remember it goes by 25% each box. So, unfortunately, not everything is just simple and normal genetics. Instead, we have some other non-Mendelian genetics, which include first our sex-linked inheritance. So this is the inheritance where your trait is found on one of the sex chromosomes. So remember, our sex chromosomes are X and Y. Females are XX, males are XY. And with these, we only really focus in this level on genes that are found on the X chromosome. So when you're looking at it, the X chromosome is the only one that's going to get letters written next to it to represent dominant and recessive. Females are going to be the only one that are able to be carriers, meaning they have a dominant and a recessive trait. Because again, the Y chromosome is completely different from the X. So it can't have those genes on it because it just doesn't have them. So it's not represented. So the Y just stays blank. They often don't ask much about that, but just keep in mind, trait goes on the X, not the Y. All right, two that I do see a lot of are incomplete dominance and codominance. When I did the review in my class, a lot of students mix these two up. More remember codominance than incomplete. So let's talk about the difference so you can not run into that error. So the main example for incomplete dominance is almost always the one I see every year used, which is the red, white, and pink flower example. So incomplete dominance, if you have a capital, so you have a trait that is considered dominant. In that case, it's usually the red or whatever it might be. Okay, so you get two capitals, you get a red flower. You get the lowercase or recessive traits, you get a white flower. However, when you get both, red's not fully dominant. It's incompletely dominant. So because it's incompletely dominant, instead of it being just red, it's going to actually blend with that white color. So it's like if you put red and white paint together, you would get pink paint. Same thing's happening here. You're getting a complete blending of those colors, so you don't get both. You just get an entirely new phenotype, an entirely new trait. Okay? Codominance is where both are actually dominant together. So instead of getting a blend, you actually just get a little bit of both. So this is showing a sickle cell traits where you get the normal blood cells, the sickle cell blood cells, and then if you're a carrier, you get both normal and sickled blood cells. Whoa. My dog just thought the guinea pigs were playing with her. When do you know when they're going? So normally when they ask questions about incomplete or codominance, I got a question in the chat. It was just to me though, for anyone who's wondering what I'm talking about. So when they do these, they often don't ask you to do a Punnett square from my experience. If they do, it will usually tell you enough to be like the heterozygous color is pink or the heterozygous gives you a black and white cow. They usually tell you that information if they're asking you to do a Punnett square with them. But usually the question is just like, we had a red flower and a white flower and we got a pink flower. Which type of dominance allows this to happen? Or we had a black cat and we had a yellow cat, and when we mixed them together, we got a calico cat. Or we had a um, black and white spotted cow. It will usually be asking you to identify the type. Okay, and then that's a good question too. So I got, is sex-linked inheritance a type of complete dominance or a different kind of dominance? So this is a type of complete dominance as well. So meaning if you have the dominant, so like down here, a female carrier that I just highlighted, that is, you have it or you don't. So she inherited the dominant and the recessive, but she's going to only show the dominant. So she's just showing complete dominance. For males, again, if you have the dominant, you're dominant. If you have the recessive, then you aren't. So that is a good question. It's just a complete dominance type. It's tricky with the X's and Y. Wait. Okay. So sex-linked isn't necessarily codominance. I wouldn't think that way. It's just normal dominance, like the one that I talked about first. Oh my gosh. I'm gonna have to lock my dog out here in a second. Sorry, she thinks that they're playing with her, the guinea pigs at least. And they're not. They're hiding. But yeah. Um she doesn't get it. Okay, where's my thing? Okay, there we go. All right, so just think sex-linked, normal dominance. The others are their different things. So again, they'll usually ask you to, if they give you the example, to say if it's co or incomplete or one of those. All right. Other things we talked about with genetic inheritance was when there's multiple alleles. The only example we talked about this year is, at least in detail, was the blood type. So blood type is identified by receptors that are on the surface of blood cells. And there are actually, well, there's only 3 alleles. There's A alleles, B alleles, and O alleles. A and B are both dominant. So this is a co-dominant situation, whereas O is recessive. So A and B are dominant, O is recessive. So if you inherit 2 A's, you obviously have A blood. If you inherit an A and an O, you have A blood because A is dominant over O. Same with B. 2 B's gives you B. A B and an O also gives you B. If you get an A and a B together, I said they're both dominant. This is an example again of codominance. So you get an entirely new type of blood called AB blood, where you have the receptors for both A and B. And then obviously if you get 2 O's, because it's recessive, that's the only way you can get O blood. So you get OO. Main thing is that it has more than 2 alleles. So it has more than just dominant or recessive. It has A, B, O. There's 3 there, okay? And then polygenic traits is not one we did Punnett squares for because that would be difficult. But this is just saying that most traits that we have actually have multiple genes that control it. So height is one I can say there's actually 6 genes that control someone's height. So that's why people and families can be such varying heights because they're inheriting 6 different genes that are determining their height. So it's not just one gene that tells you everything about your height. There's a bunch going on there. And so there's going to be many different influences in those situations. Same with skin color, eye color. It's a lot more complicated than just looking at one gene. Okay, keep chugging along here. It's going to be a little bit longer than the half an hour, but that's okay. All right, so now we talked about how you can inherit genes, but we need to talk about what actually makes your genes visible and possible. So we already talked about how DNA copies. So now we just want to talk about the 2 steps to creating ultimately the protein, which is going to be what allows traits to be shown. So protein synthesis starts with the process of transcription. Transcription happens in the nucleus because that is where the DNA is. DNA does not leave the nucleus ever, okay? It can't get out. So DNA, because it's double-sided, okay, it can't get out, which is why we create the next molecule, which is mRNA or messenger RNA. Remember, RNA is single-stranded, and it's actually going to be able to fit through the holes in the nuclear membrane so that it can leave that location. Oh my gosh. Hold on. Now I set the guinea pigs off. Um, all right. So when you're creating RNA, remember there is a slight rule change. I really hope you can hear the guinea pigs right now. They are squeaking so loud. Um, I need to move rooms. So during transcription, instead of it being T to A to T, C to G, okay, there is that change where the U comes in. So T is replaced by U. So obviously the DNA is still going to have T's. So T is going to give you an A still because they still exist. But anytime you have an A now, instead of getting a T, you get a U. C and G remain the same. So they're still paired up. Nothing changes there. So just with that, remember that a U is now formed. For translation, remember this takes place in the ribosome. Okay. And this is where the proteins are made. I say if you pronounce ribosome like rib, think ribs. If you eat ribs, those are giving you protein. Okay, so proteins are made in the ribosome or ribosome. Okay, with that, it's kind of like a factory. So the mRNA comes into the factory, and the tRNAs are going to carry amino acids in to match up with the codons. So a codon is a group of 3 bases. Okay. Oh, I'm sorry. Um, the codons, again, are groups of 3 bases. tRNA is going to have an anticodon, which is the opposite. So if it's an AUG, you get UAC, and so on. Again, they're going to carry in the amino acids, which amino acids are the building blocks of proteins. So that is how the code actually tells the protein how to be built. So those amino acids are going to link together until the mRNA goes completely through, and then it's going to release the polypeptide, and that polypeptide is going to fold and create a protein. My animals are noisy tonight. So transcription, translation. That's the order. It goes alphabetical. So a C comes before an L. Okay, so it goes in order that way. Oh my gosh. Hold on. I gotta lock them out. I'm just gonna hold her in jail by holding on to her. Okay. So not often they ask you to do this, but they might. So when you are actually trying to take RNA and turn it into a codon, or an amino acid, sorry, you are going to use a codon chart. So if you started and you have UCA, okay, on this chart, you just go first, second, third. So U is the first one. Oh, it's not going to let me show you that. C is the second one, and then A. So your amino acid would be serine or ser. If you're using a circle chart, you start in the center. Say I gave you UAC, you go just out. So big, medium, small, and you get tyrosine. I'm not too concerned about the mutations, but in a change for DNA, there are point mutations where one base is changed. There are insertions where you're inserting a base. Okay, so that one's pretty self-explanatory. Deletions where you're deleting or removing a base. Words that are used to describe a change in the protein is a missense mutation where there's one mistake, okay? One amino acid is affected. There's a nonsense mutation where basically the whole molecule is stopped early, okay? So an entire protein isn't even built. A silent mutation is basically where nothing changes in the protein. So even though the DNA was affected, sometimes there are similar. So you can see here like CUU and CUA both give you leucine. So if the mutation occurred in that way, it would just give you the same thing anyways. So it's silent. Frame shift is really bad. So when you're inserting or deleting entire sequences, it's going to mess up every amino acid after the change. Chromosomes, I'm even less worried about them asking you, but duplication, it copies the section, it duplicates it. Inversion, it basically just flips it, so the DNA is read in the wrong order. Deletion, deletes a section. Insertion, again, inserts a section. And then translocation is basically crossing over, where two chromosomes are going to trade sections of DNA, but this time the sections are between two completely different chromosomes. So it's completely different, I just said different twice, completely different DNA, meaning everything just gets messed up from there. All right. We made it to evolution. So evolution and ecology, I'm going to move pretty fast through just because those are the two most recent. And these are the questions that students always score the highest on, even though we spend the least amount of time on them. Okay, so remember theory of evolution. Quick recap. A theory is a generally accepted hypothesis to how something happens. Okay, and evolution is going to be a change of a species over time. So from generation to generation, a species changing, and it's not often a quick change unless it's an organism that reproduces really, really, really fast, like bacteria. This is something that takes centuries to happen. So macroevolution would be something that takes that long time, whereas micro is looking at small changes that happen in smaller amounts of time. So things that can cause evolution, there's natural selection, which is the process of nature essentially choosing the organisms that are best fit to survive or best or most likely to survive. So I know most classes look at the moth example, where the moth that was able to camouflage better was the one that survived better and therefore the one that became most of the population later on. This image is always so sad. Students always don't like this one. Okay, but there was apparently giraffes once upon a time that had shorter necks. Well, as the trees in the area were taller and they couldn't reach their food, all the ones with the shorter necks died off so that all giraffes had longer necks so that they could reach their food source. Sexual selection is a type where, based on the traits that are favored, within a population by reproductive partners. Those are going to be the traits that survive. And then selective breeding or artificial selection is where specifically humans choose the traits that they want to basically influence the outcome of further generations. So this happens a lot with crops, happens a lot with pets, and things like that, where we are choosing the outcome of the generations instead of the animal themselves. Um, Natural selection can take different forms. So there can be directional selection, where one extreme is chosen as the favorable trait. There's stabilizing selection, where just kind of the intermediate trait, the trait that was kind of in the middle of all of the other ones, is the one that is selected. And then disruptive selection is where the two extremes are chosen. So let's say the giraffe example. Let's say we had short, medium, and tall necks. I don't know. This one would be where like the short and the tall both survive, and the medium would not. Okay. And then our evidence, or actually, let me do this first. More mechanisms of evolution. So the main thing that causes evolution to occur in the first place would be mutations. So the random acquiring of a new trait that's going to possibly be beneficial to a population. There's genetic drift, where because of something like a weather event or a natural disaster or a bunch of organisms leaving or illness or whatever it might be, the allele frequency in a population is going to change. So you might have more of a trait than you did before, and therefore that affects how it gets passed down. Founder effect is when a small population or a small number of individuals moves from a large population to a new area where there is no one of their species. So they are starting the new population. Bottleneck effect is when there is a sudden large reduction. So this is again like illness or like a natural disaster where a large amount of the population is wiped out. And so whatever's left are the organisms that pass their traits on. And then migration, so organisms that move or into or out of an area are going to affect a population. Evidence of evolution, there's geography, so obviously land, if they're split up by land. Um, we can track what differences there are. So we used to have Pangea, and we can see organisms that would have used to live near each other that no longer do, can be seen often as similar. You can also just see, like within the Galapagos Islands is the main example where this all started, is that each island had similar organisms but different variations based on the resources on each island. So geography, there's homologous structures where the structures themselves are similar in look or just structure in general, but the function is different. So a human arm, a horse foot, a bat wing, and a porpoise fin all have similar bone structures. However, they all have different functions. So that usually indicates a common ancestor. Embryology is basically just saying that embryos on early stages of development look very similar to each other and therefore could indicate, again, a common ancestor. Vestigial structures are structures that no longer have a function to the species. So a whale having leg bones. Whales don't have legs, and the bones are just kind of floating in its abdomen. So not a structure that's needed anymore, but indicates that a whale at one point might have had legs. Analogous structures are basically the opposite of homologous. So that's where we get similar functions now, but the structure is completely different. So like a bat wing, is that what? Oh, that's a bird wing. Bird wing, a bat wing, and a butterfly wing all are completely different in structure, but they all have the function of flying. Okay, so they evolved independently of one another. And then comparisons in DNA and proteins, we can take DNA and proteins because there is not a wide range of those. and compare them to see how closely related two species are to each other. Gradualism and punctuated equilibrium just basically states how changes occurred over time. So gradualism is there were small changes over a long period of time, whereas punctuated equilibrium is that there were bigger changes over short periods of time. And then we covered that. Okay, and then the last one, ecology. This was the last unit that we did. So ecology is the study of organisms and interactions with each other and their environment. So starting kind of big here, we have a biome. A biome is just an area that is classified by its climate or the plant and animal species that are present in that environment. Okay, so we have forests, we have deserts, grasslands, tundra, rainforest, deciduous forest, all those. Then we also have our aquatic biomes like the ocean and freshwater biomes. Let me see. Okay. These two slides are the more important ones, I believe, though. So succession is a predictable but long process of how an ecosystem rebuilds over time. So there's two types. There's primary succession, which is when there is no life at the start of the process. So it's just barren rock, which again, this usually happens after a landslide where rock is completely covering the ground, or a volcano where molten rock forms a new environment. And this takes, again, thousands upon thousands of years, but eventually pioneer species like mosses come in, break down the rock into soil, and then once the soil is formed, small plants can grow until more and more soil forms, and eventually larger species can grow. Secondary means, I always say S for soil. So wildfire is the biggest example. Forest burns down, but there's still soil, there's still seeds laying around. So small plants can start to build again until we eventually grow our larger species again. Um, but competition. So I think you all know what competition is. So fighting for the same resources, whether that's food, habitat, water, mates. That's all part of that. Carrying capacity is what happens when an environment basically reaches the maximum number of organisms it can support. So there's not unlimited resources in any situation. So organisms can often grow and grow and grow and grow and grow in a certain area until they reach, maybe they run out of food. There's not enough rabbits to hunt based on this picture. So this kind of line where it's going to fluctuate up and down around it is going to say this is the maximum number of lynx, okay, this animal here that we can support because of the rabbit population. A limiting factor is basically what I just said. So it's something that limits a population growing forever. So it's the thing that literally causes the carrying capacity. So again, the snowshoe hare population is a limiting factor to the population size of the lynx. Limiting factor could also just be illnesses, how fast an organism reproduces, anything like that. Okay. And then the last slide. So symbiotic relationships. So symbiotic just is an interaction between two different organisms where one is going to benefit. So mutualism is the one where it's all happy. Both organisms benefit from this relationship. Commensalism is where one organism, as I said, always benefits. So one benefits, and then one is neither helped nor harmed. So it's kind of just neutral. And then parasitism, one organism benefits while the other is harmed. You don't need to know this anymore. They took that out. Non-native species are species that don't belong to a certain area. Non-native could be good or bad to an area. That just means they're not from there. We usually bring in the term invasive species when they are harmful to the area in which they become part of. Okay. And then we have food webs and food chains. Food webs, remember, are a lot more complex and they can show multiple organisms and how they all interact with each other. Whereas a food chain, it goes one to the next to the next to the next. So it's only showing one path, whereas a food web's kind of like a choose your own path kind of situation. Um, Remember the key words here. Producers are the ones that produce food from sunlight, so usually are plants, okay? And then we have primary, secondary, and tertiary consumers. So primary consumers are the ones that eat the plants. Secondary consumers eat the primary consumers. And then tertiary eat the secondary. So it just is going up the line. Remember, as you move through a food chain, energy is lost. So only 10% of the energy makes it to the next level. So if we're starting 100% at my plant, okay, you're going to have only 10% by the time you get to the bug. And then you're only going to have 1% by the time you get to the blue jay. And then you're only going to have 0.1% left by the time you get to the lynx. So a lot of energy is lost through things like body heat, waste, anything along those lines. And finally, I would just know the basis of the cycles. So I don't think you need to know everything. So water cycle, I would say they often ask the most about how runoff can affect like pollution. So runoff is the stage where water, once on the ground, can flow across the surfaces like roads or soil, and it can collect anything that's in on those surfaces and put it into the waterways, which could pollute them. Um, Carbon cycle, this is where, okay, we take the only way carbon exits the atmosphere is through plants and photosynthesis. So photosynthesis is the only thing that removes carbon. Carbon is then broken down as plants and animals die and put into the soil. Over generations, fossil fuels are formed, and we use those fossil fuels as fuel for factories and cars. Okay, and so we're putting that carbon back into the atmosphere. Problem with that is the plants aren't keeping up with the amount that we're putting into the atmosphere, so the atmosphere has too much, which is the issue we're seeing related to the carbon cycle right now. Nitrogen, main thing I'd know is just that there are bacteria involved. All right. A bit longer today, but that's again okay. Um, okay. I just got a question. Sorry, I'm reading it. Um, so it says an ecological organization, how do you know which ones are defined as living, non-living, or climate? Um, so one thing, remember the words for living and non-living that also could be used are biotic and abiotic. Um, so living things would just be any of the plants or animals, whereas non-living would also be tied to climate, if that's what you're asking. So, like anything that's not a plant or animal is considered abiotic. So, rain, rocks, actual water on the ground, soil, any of that would be considered non-living. Let me know if you need further clarification there um, on that. Let's stop sharing. So yes, at this time, I'm open to any questions. Um, if not, you've been here for a while too, so if you would like to start hopping off, you're welcome to do that as well. But I'll kind of stick around for a few minutes to make sure I answer anything you need. Water properties. They will not ask about the water properties because that would have been on Mondays because that was the chemistry unit. So I'm guessing they didn't ask about the water properties then. On the study guide, there was a question about whether the biome ecosystem. Oh, let me see if I can figure out what question you're asking me about. Oh, I cannot minimize the zoom. Okay. Oh, you said study guide. Not that one. Oh, okay. Let me pull that up. That would be at the end. I apparently don't have that one on here. So I would just basically climate. I don't think they're going to ask you that on the Keystone. But I can't find it to pull it up that quickly. The document is missing. But basically, again, living would be any of the plants or animals in a biome would be living. Non-living would be like rocks, soil, water, anything that like is in. So like if I'm describing a desert biome, the living things would be the cacti and the camels and the snakes, whereas the non-living would be the sand and there's not much else, rocks usually. And then the climate would be describing the temperature or the amount of rain that happens. So the weather stuff is the stuff that would be climate. Um, Again, I don't think they're going to ask that one directly, but it's definitely important to know the difference between just biotic living or abiotic. The things we did on Blookit and Gimkit. Aubrey, I think I'm confused by your message. No, you're good. Um, I just wanted to make sure I could clarify the right thing for you. Okay. The things we did on Blookit and Gimkit are the vocab terms for the Keystone, yes. I think that's what you're asking.